Well, thank you, Dr. Hobson, for sharing your work on correction of PLP1 splicing. My name is Paul Tizar, and I'm a professor at Case Western Reserve University in the School of Medicine. I'm also the chair of the Scientific Advisory Board for the PMD Foundation. So I will be your next speaker, and I'm really excited to share recent work from my laboratory on a new therapeutic approach to knock out PMD. So I'll start here just giving you a quick overview on this first slide of the approach that my lab has taken to advancing new therapeutics for disorders of myelin. And so we're really committed to advancing the discoveries in our laboratory into clinical testing for patients with myelin disorders. And over the last 10 years, we've really developed new stem cell-based tools that allow us to understand the underlying processes of neurological disorders, including Pelizaeus-Merzbacher disease. And the goal, of course, is to develop new medicines that we can bring to patients. Now, we've really spent a lot of time since the lab started 10 years ago focusing our efforts on multiple sclerosis. We've built new tools that allowed us to understand this disease, and we've developed new potential therapeutics that have now reached the commercial sector in a startup company that I co-founded called Convelo Therapeutics that's now partnered with Genentech to advance these forward into clinical testing for MS patients. And so what I want to share with you today is how we're really using and leveraging the expertise and technologies that we have in the laboratory for Pelizaeus-Merzbacher disease. And hopefully at the end of this talk, you'll share my excitement that we have a potentially meaningful and impactful therapeutic option for patients in the not too distant future. And so I just want to start here not only by thanking the PMD Foundation for hosting this conference, as well as the sponsors and CHOP for all of their hard work and organization, but to the families in the foundation specifically. It was in 2013 that I received a small pilot grant from the PMD Foundation. I was a relatively new assistant professor at the time, still trying to sort out exactly what I wanted my lab to focus on. And this grant really cemented our long-term interest in PMD, and it was a few years later where I was able to join and meet some of you at the family conference that really allowed us to pursue the work that I'm going to share with you today. So hopefully, you all feel that you've committed a big part of the work that I'm going to show you, and together, clinicians, scientists, as well as patients and their families are making a difference. I think we've really reached a new era. Hopefully you agree, seeing some of the scientific talks today, that we're really moving forward with new potential meaningful therapies for patients. So as you've all heard, oligodendrocytes are important. So on this slide here with this brain, you can see in the middle in this cartoon in blue, the oligodendrocyte. And of course, you know that these cells physically reach out and wrap around the nerve cells to create this insulating and protective coating called myelin. And in what this myelin does, as well as some of the other important functions of oligodendrocytes, is to support the nerve cells and allow them to function properly. And we know that when these oligodendrocytes don't provide these functions, that the nerve cells can't transmit electrical signals appropriately and become dysfunctional, and this leads to patient disability and disease. Right? And so there's a class of cells called oligodendrocyte progenitor cells whose normal job, as you've heard, is to make new oligodendrocytes. But in PMD, that whole process becomes faulty. So you have genetic mutations in PLP1 that cause the oligodendrocytes to either die or be dysfunctional. And the oligodendrocyte progenitor cells also carry those same genetic mutations and therefore can't make healthy oligodendrocytes. And so what I'm showing you here on this next slide of a movie, on the left with healthy and on the right with PMD, is I told you we've developed these tools in my laboratory to allow us to study the cells that are normally found in the brains of PMD mice and PMD patients, but we can now study them and grow them in a petri dish in the laboratory. So in this movie, you'll see the healthy cells on the left develop these very beautiful spider web-like oligodendrocytes. But on the right, you'll see that the PMD cells, most of them just sort of poof and die throughout the culture. And so what this means is that it's really challenging. We face really a difficult task, is how do we keep these oligodendrocytes alive, and how do we allow them to provide supportive function to PMD patients to sort of prevent or reverse paralysis and allow normal neurological function. And so on this next slide here, back to the brain cartoon, this was really the question that I asked. And so how do we rescue oligodendrocytes in Pelizaeus-Merzbacher disease? And that's really what this scientific session has been about, and you'll continue to hear from speakers after me. All right, so we've heard about stem cell and progenitor cell transplantation as a promising strategy where you put otherwise healthy cells and you physically transplant them into the brain of patients. And those cells could then generate healthy, normal oligodendrocytes and potentially provide enhanced neurological function to patients. A different option would be to not do transplantation, but moreover to provide a drug therapy where you're providing a supportive function to the mutant cells. And so you're allowing them to potentially stay alive or decrease their stress to enhance their function. And you'll hear about this either through diet modulation or other drug therapeutics from speakers after me. And what I'm going to talk to you about today is a genetic type therapy, really going after the root cause of PMD itself, which is the genetic mutations in PLP1 gene. So on the next slide here, what I'm showing you on the left is the DNA. So this is the genetic code of life, and these individual letters make up all of our genomes. And in the PLP1 gene, here what I'm just showing you in this cartoon is just indicating a healthy genetic sequence. And DNA makes an RNA transcript that then codes for the proteins within a cell. And in the case of PLP1, a normal PLP1 DNA sequence makes normal PLP1 RNA, normal PLP protein, and that allows us to have functional oligodendrocytes on the right. But on the bottom here in PMD, we have mutations in the PLP1 gene that cause mutant or excessive PLP1 RNA, which causes mutant or excessive PLP1 protein, which is then toxic to those oligodendrocytes and causing them to die here, indicated by the loss on the lower right. So in the next slide, the approach in my laboratory, quite simply, was could we go directly after these PLP1 mutations using a new class of technology called CRISPR-Cas9. So this CRISPR technology, you can think about it as sort of DNA scissors, indicated here in the picture on the lower left. And the question is, could we go after the mutations directly in the DNA, cut them out, and then that would prevent the formation of mutant RNA, which would prevent the formation of the toxic PLP protein and possibly restore functional oligodendrocytes on the right. And so I just want to use this next transition slide to quickly introduce you to the animal model of PMD that we have focused on in my laboratory, which is the Gympie mouse. Gympie is a severe model of PMD. It models the most severe form of PMD, the connatal form. It causes a mutant PLP protein to form in the cells, which kills oligodendrocytes. You can see this here in the lower left, where you have a cross section through the brains of healthy or Gympie, these PMD mutant mice. In green is oligodendrocytes or myelin. So you can see in the wild type or healthy animal on the top, you have lots of oligodendrocytes and myelin in green, whereas in the Gympie animal, it's very few at 19 days of age. In this video here in the middle, you can see the healthy animal on the right, whereas the severe Gympie animal has intention tremor and ataxia. And unfortunately, these mice succumb to their disease within 3 weeks of being born, where normal mice, healthy mice, will live over a year of age. What we did was we used these PMD mice, these Gympie mice, and asked if we use this CRISPR DNA molecular scissors technology, could we go in and knock out or cut out the PLP1 gene and prevent formation of the toxic RNA and protein. And so on the top, there's the healthy animal. On the bottom is the PMD animal, and you're looking at green in myelin of the brain. And I'll pop up here the CRISPR-treated PMD mouse. you can see has a restoration in green of the oligodendrocytes at 19 days of age. Now, just as a reminder, these mice normally die at 21 days of age on the bottom. And quite excitingly, at 6 months of age, this CRISPR treatment of these mice, where we've knocked out the PLP1 gene, allows these mice to grow into adulthood and to have almost indistinguishable amounts of oligodendrocytes and myelin from the healthy animals. And I'll show you that more directly here in this video on the next slide. On the left is the CRISPR-treated animal, and on the right is the PMD animal, and you can see that the CRISPR treatment allows these mice to move around normally, whereas the PMD mouse has quite severe intention tremor, ataxia, and seizures. And so this is a complicated graph, but in what I just want to summarize is in red, those PMD mice normally drop off and die at 3 weeks of age, whereas the CRISPR-treated animals live out past 18 months of age, a normal lifespan. And so this was quite exciting to us, and what we showed is that CRISPR knockout of PLP1 was feasible and well tolerated in mice, and that CRISPR knockout of PLP1 really provided sustained restoration of oligodendrocytes, myelin, neural function, locomotor activity, motor coordination, and importantly, full lifespan extension for these animals. So it was quite remarkable and quite exciting. And so CRISPR inactivation of the PLP1 gene could provide a future therapeutic option for PMD patients. But I think importantly to note is that CRISPR technology is quite new, and although quite exciting, it's not currently in use for human neurological disorders. And the ability to deliver the CRISPR machinery to oligodendrocytes is really not currently feasible. And so we wanted to, after we showed that this was a really exciting approach to knock out PLP1, we wanted to ask, were there therapeutically relevant ways that we might be able to do this? And so we turned to a class of drugs called antisense oligonucleotides, or ASOs, and you've heard about these in the previous talk from Grace Hobson. And just as a reminder, the question here was, could we use these ASOs to not go after the DNA, but to go after the RNA here in green, and to suppress or knock out the expression of that RNA, which would prevent the expression of the toxic proteins, which would then potentially allow for restoration of functional oligodendrocytes. And so as you heard, these antisense oligonucleotide drugs, these are sort of small pieces of DNA, the sort of building blocks of our genetic code. They can be specifically made to target any gene or RNA in the genome, and in this case, PLP1. And so you can make them, they can target and suppress the RNA expression, and then reduce toxic protein formation. And so the exciting thing about ASOs in comparison to CRISPR technology is that ASOs are already approved for human use for one neurological disorder in particular, spinal muscular atrophy. Nusinersen has become a blockbuster drug for Biogen and Ionis. And there's a number of clinical trials for other disorders of the nervous system ongoing. So these drugs are delivered directly to the nervous system. And so it allows us to specifically target knocking down or knocking out a particular toxic RNA and protein in the nervous system. And so I'm just going to end with a few slides on a very recent study that we just published earlier this month in Nature. And this was a collaboration with Ionis Pharmaceuticals, in particular with Barrett Powers and Frank Rigo, who worked with us on this study, where we've utilized antisense oligonucleotides targeting the PLP1 gene in PMD mice. And so you can see here in the next slide where we're really looking at these green images of cross sections of the brains of animals. And so I'll just walk you through this quickly. The top row is animals at 3 weeks of age. The bottom row is animals at 8 months of age. And then the top row on the left is the healthy or wild type animals. On the right is the GIMP or PMD mutant animals. And you can see the mutant animals have very little green, so very little myelin. And so in the middle, what you're seeing is two different ASO drugs that we've treated these PMD GIMP mice with. And you can see very nicely a nice restoration in green in the middle of these myelinating oligodendrocytes that's sustained way out through 8 months of age in the bottom row. And so in this video here on the next slide, in the upper left is the healthy or wild type animal. In the upper right is the mutant PMD or GIMP animal. And the two treated GIMP animals or PMD animals with the ASOs are in the bottom two videos. And I'll play this now, and you can see that in the bottom two videos, the treated mice can move around their cages normally, whereas in the upper right, the PMD or GIMP mouse again has quite severe phenotype, and those mice die at 3 weeks of age. And so excitingly, when we treat these PMD or GIMP animals with these ASOs or antisense oligonucleotides targeting PLP1, we can show that they can have sustained recovery into adulthood. So on the bottom are PMD mice out at 6 months of age that were treated with these PLP1 antisense oligonucleotides. And this graph, again, is complicated, but to summarize, on the left in red is showing that these GIMP or PMD mutant animals drop off or die at about 3 weeks of age. But in the top, the two blue lines show that when we treat those animals with antisense oligonucleotide drugs targeting PLP1, we can extend their lifespan out past 8 months of age. And so I'll conclude there, really just showing you that these antisense oligonucleotide drugs can efficiently be delivered to oligodendrocytes in the central nervous system of animals. We've shown that we can use them to suppress PLP1 RNA and PLP protein for extended durations. We've shown that these ASO-mediated suppression of PLP1 restores oligodendrocytes, restores myelin, restores function, neurological function, and quite excitingly has a quite dramatic extension of lifespan in these PMD mice that otherwise succumb to their disease at 3 weeks of age. And so we're really excited about this because as ASOs are already approved for human use in other neurological disorders, there's really a clear path forward for PLP1 ASOs to potentially be advanced into clinical testing for PMD patients in the not too distant future. And so I'll end there and just thank the team. This was a tremendous amount of work across a large number of individuals. I'll highlight Matt Elit and Lane Barber, who led this work in my lab with support from a large number of individuals, and in particular at Ionis, Barrett Powers and Frank Rigo. And of course, none of this would be possible without funding, and it was the initial seed grant from the PMD Foundation that really allowed this work to move forward. And so with that, thank you very much for listening today. I'm really excited to be able to share this work, and please reach out to me if you have any questions.